So, good morning. Let's see about the gluconeogenesis and we will see how it's contrasts with glycolysis. Gluconeogenesis is a process by which the glucose molecules are produced from the non-carbohydrate precursor or substrates. These are the non-carbohydrate substrates: lactate, glucogenic amino acid, glycerol, propionic acid, and out of these, the lactate and glucogenic amino acid are the most important and most frequent substrate for the glucose synthesis. And what is the site? Site is basically the liver, and to a very less extent, it happens in renal cortex also. The pathway is partly a mitochondrial and partly cytoplasmic. So, what is the significance of gluconeogenesis? There is a vast role of gluconeogenesis in maintaining the homeostasis of blood glucose. So, the brain, RBCs, lens, and cornea of eye, testes, kidney, medulla are dependent on glucose for continuous supply of energy. Brain has minimum need of 120 grams of glucose per day. So all time there should be a minimum concentration of glucose in the blood should be available in order to provide them the continuous supply of glucose. The body stores of glucose and glycogen are depleted within a first 12 to 18 hours of fasting. So, in the fasting or starvation, even more than a day, gluconeogenesis must occur to meet the basal requirement of the body for glucose, which is essential for the survival. Certain metabolites accumulates in the blood, like lactates produced by the muscles during vigorous or strenuous exercise, and RBCs also, and glycerol produced by the adipose tissues, all these to be scavenged and converts up into the useful form via gluconeogenesis. Then increased gluconeogenesis is observed in critically ill patients. So, in nutshell, significance of gluconeogenesis: it maintains the blood glucose homeostasis in lack of carbohydrate in the diet, ensures a continuous supply of glucose to the tissues which require a continuous supply of glucose, such as brain, RBCs, lens, cornea of the eye, and kidney, medulla. And it clears the products of metabolism of other tissues from the blood, like lactate and glycerols. Then gluconeogenesis is not just the complete reversal of glycolysis. There are three reversible reactions of glycolysis are catalyzed by hexokinase, phosphofructokinase, and pyruvate kinase. So these three stages are catalyzed by four alternate enzymes specific to gluconeogenesis. So definition wise, the glycogenesis is conversion of from glucose to pyruvate via these steps: from glucose to glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-phosphate, and phosphoenol pyruvate, and finally to the pyruvate. So this is glycolysis. While in gluconeogenesis, pyruvate is one important substrate which reverts back to phosphoenol pyruvate, and phosphoenol pyruvate to fructose 1,6-phosphate and fructose 6-phosphate, glucose 6-phosphate, and finally to the glucose. So new synthesis of glucose from the non-carbohydrate substrates. But it's not a complete reversal of glycolysis due to the differences in the enzyme which carrying out the steps in reverse direction. So we will see the line of difference basically between the glycolysis and gluconeogenesis. It's a very well known first step of glycolysis is glucose to glucose 6-phosphate, which is being mediated by by hexokinase. So in gluconeogenesis, this is being compensated by glucose 6-phosphatase. Means if you wish to synthesize glucose 6-phosphate to be converted to glucose, so in reverse direction, we need glucose 6-phosphatase against in contrast to hexokinase. Similarly, the second enzyme, the difference of second enzyme means fructose 1,6-phosphate has to be reverted back to a fructose 6-phosphate in gluconeogenesis step. So the enzyme is fructose 1,6-bisphosphate, which is working in contrast to phosphofructokinase 1 enzyme of the glycolysis, which converts fructose 6-phosphate to fructose 1,6-bisphosphate. The third difference of enzyme between the glycolysis and gluconeogenesis is there. In glycolysis, the pyruvate kinase, in simple one step, it converts phosphoenol pyruvate to pyruvate. While we wish to return to phosphoenol pyruvate during gluconeogenesis, so there are two compensatory enzymes which are working in contrast to pyruvate kinase. One is the pyruvate carboxylase, which converts pyruvate to oxaloacetate, and from oxaloacetate to finally phosphoenol pyruvate. So that is mediated by the enzyme phosphoenol pyruvate carboxykinase. So one enzyme of the glycolysis is no compensated by the two enzyme in the reverse direction in the gluconeogenesis. One is pyruvate carboxylase, one is the phosphoenol pyruvate carboxykinase. So this is how the reversal of glycolysis is happening. Most of the steps, and the enzymes involved in the various steps, they are same, but these are the three major steps which are being compensated by three different enzymes in the gluconeogenesis. So more clear account of the gluconeogenesis and glycolysis. So let's see, we have already seen about the glycolysis, a very important pathway of generation of pyruvate or lactate right from glucose. So you can see it here, the glucose to glucose 6-phosphate, the hexokinase is there, or sometimes glucokinase. So glucose 6-phosphate to fructose 6-phosphate mediated by then fructose 6-phosphate to fructose 1,6-phosphate, then fructose 1,6-bisphosphate mediated by aldolase enzyme. It converts or broken up into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and then 1,3-bisphosphoglycerate, then to 3-bisphosphate glycerate to 2-phosphoglycerate, and finally 2-phosphoglycerate converts to phosphoenol pyruvate, and finally via pyruvate kinase it converts to pyruvate, and pyruvate goes into the mitochondria. It converts up into oxaloacetate via the enzyme pyruvate carboxylase, and this is the oxaloacetate, and this is the Krebs cycle. This is one of the important intermediate of the Krebs cycle, oxaloacetate, and then it finally converts up into the via the citrate and isocitrate. It converts up into the alpha ketoglutarate, then succinyl coenzyme A, then fumarate, and finally to the malate. Then malate comes out because now let's talk about the reversal of glycolysis, that is the gluconeogenesis. The major substrates we keep into account. One is the pyruvate, one is the lactate is there. If there are anaerobic conditions, so pyruvate, instead of pyruvate, it is the lactate, which is the major product, end product of the glycolysis, that is also involved in the gluconeogenesis. And then we'll see the various glucogenic amino acids, how they are involved in the synthesis of glucose. Then we'll see how the glycerol is involved in the gluconeogenesis. Glycerol is produced as a byproduct in various adipose tissues. So one by one we'll discuss. So you can see it here, the malate. Once we are in the fasting, in vigorous fasting after 12 hours, because most of the sources of glycogens are over during the fasting. After 12 hours, it's the gluconeogenesis come into the play in order to maintain the normal level of blood glucose, because few organs, as I've already told you, like brain and renal cortex and certain other RBCs, they all require continuous supply of glucose. So body never know whether we have eaten or we have not eaten, but they want glucose supply all the time. So in order to maintain the homeostasis, the malate is going to play an important role. Oxaloacetate, you can see this is the reversible step. So oxaloacetate to malate or any of the intermediate of Craig cycle finally converts to malate, and from malate to malate because it can easily pass through or cross over the barriers of mitochondrial membrane. It comes into the cytosol. In the cytosol, again the malate is converts up into the oxaloacetate. Here the NAD is converts to NADH, and this is the malate dehydrogenase enzyme. Then oxaloacetate finally converts up into phosphoenol pyruvate. The enzyme is phosphoenol pyruvate carboxykinase. So two important enzymes of the gluconeogenesis from one is the pyruvate carboxylase and one is the phosphoenol pyruvate carboxykinase. They are mediating the reverse conversion of a pyruvate to phosphoenol pyruvate, which only required pyruvate kinase in glycolysis in order to convert to PEP to pyruvate. So this is the major difference. Then further, the phosphoenol pyruvate follows the same step, reverse steps of glycolysis. The same enzyme, it converts up into the 2-phosphoglycerate, from 2-phosphoglycerate to 3-phosphoglycerate, finally to 1,3-bisphosphoglycerate, and finally to from glyceraldehyde 3-phosphate, it converts up into the fructose 1,6-bisphosphate. Further, fructose 1,6-bisphosphate is converting back to fructose 6-phosphate. Here, the second difference comes the phosphofructokinase, which was working for conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Now, in the reverse direction, if in the gluconeogenesis pathway, the enzyme is fructose 1,6-bisphosphatase. So it is reverting back glucose 1,6-bisphosphate to fructose 6-phosphate, right? Then the next difference is now further through isomerization, fructose 6-phosphate converts up into the glucose 6-phosphate, the same enzyme. Then further, finally, the last step, the glucose 6-phosphate has to convert to glucose because newer synthesis of glucose has to take place. So here there is a difference of another enzyme, that is the in glycolysis, if glucose has to convert back to glucose 6-phosphate, the enzyme was hexokinase or glucokinase, while in the gluconeogenesis, the enzyme is glucose 6-phosphatase, which finally releases the glucose. So this is the third line of difference. So how the lactate is acting as a substrate for gluconeogenesis, we can show it here. See this reaction during the anaerobic conditions. Lot of synthesis of lactate happens during the strenuous exercise, or RBCs, if oxygen lack is there. So lactate is the major product. So lactate has, it's just an end product of, or now it lies at the dead end of the glycolysis. So it has to revert or reconverted back to the pyruvate. So pyruvate, once the pyruvate is formed, again following the back steps, all these steps from PEP to 2-phosphoglycerate, 3-phospho, and finally it joins the gluconeogenesis pathway. This is how the lactate and pyruvate are involved in uh, converting to glucose. Then how the glycerol is acting as an substrate for gluconeogenesis. So lot of glycerol is being generated by the adipose tissues. These glycerol has what can be the fate, how it joins the gluconeogenesis. So you can see it here, the glycerol via the enzyme glycerol kinase, it is converted to glycerol 3-phosphate. From glycerol 3-phosphate, it converts up into the dihydroxyacetone phosphate via the enzyme the glycerol 3-phosphate dehydrogenase, and finally it joins the gluconeogenesis pathway, and this is how it is involved in the gluconeogenesis, right? Then we will see ahead or further how the propionic acid is involved in the synthesis of glucose, because propionic acid is the end product of the fatty acid or chain fatty acid beta oxidation. We'll see it clear there. Now this is the diagram showing the various glucogenic and amino acid, how they are involved in the production of glucose. So you can see it here, the serine, the glycine, cysteine, and threonine. In conditions of now, when the pyruvate is in deficiency or in lack, or there is a need of, synthesis of glucose, gluconeogenesis is to be prompted. So these can be converted up into the pyruvate. These are glucogenic amino acid, major glucogenic amino acids. So these are converting to pyruvate. Then via pyruvate, they follows the pathway of gluconeogenesis, and that's how they are involved in gluconeogenesis. Similarly, all the intermediates, they are directly or indirectly involved in the gluconeogenesis step via converting finally to malate. So you can see it here, the phenylalanine tyrosine it's convertible to fumarate. Then these valine, isoleucine, and methionine, these are all glucogenic amino acids. So they convert to succinyl coenzyme. Then histidine, arginine, and proline, they are convertible to alpha ketoglutarate, glutamate. It is the keto acid of glutamate. Then glutamate also via glutamate is convertible to alpha ketoglutarate. So you can see the volley of chains of reactions happens. All glucogenic amino acid except for leucine. Leucine is purely ketogenic. All rest of the amino acids are convertible to any of the intermediates of the Krebs cycle, and this is how they are involved in the gluconeogenesis. Now, energy requirement. So actually the conversion of 2-pyruvate to glucose, it requires 6 ATPs. So where there is an consumption of ATPs, the first step is the pyruvate carboxylase, where there is a conversion of pyruvate to oxaloacetate. So in this step, there is a consumption of 1 ATP. Then phosphoenol pyruvate carboxylase enzyme, which is responsible for conversion of that oxaloacetate into the cytosol to be converted to phosphoenol pyruvate. So this carboxylase enzyme also consumes 1 ATP here. Then third is the phosphoglycerate kinase. So this is the enzyme of Krebs cycle. This is the enzyme of glycolysis as well as the gluconeogenesis. Here also there is a consumption of 1 ATP. So overall, from 1 pyruvate to half, 3 ATPs are required for production of half molecule of glucose from 1 pyruvate. So for 1 glucose molecule, we definitely require 6 ATPs. So total energetics is the 6 ATPs consumed while converting the 2 pyruvate to glucose. Then there is a more clear vision of, lactates, how they are involved in the synthesis of glucose via gluconeogenesis. And this very, special pathway is known as a CORI, being named on the basis of discoverer. So actually what is happen in the muscle, specifically in the muscles, but organs is also can generate this lactate. So this glucose to pyruvate and pyruvate to lactate during some heavy muscle activity, and this lactate keep accumulating in the muscles. It is to be scavenged out and converted up into the some useful product, and this process cannot happen in the muscle itself. So lactate has to be transported to liver via bloodstream. So lactate reaches into the liver, and lactate converts to pyruvate, reverts back to pyruvate. From pyruvate via the gluconeogenesis, it is converting to glucose. Consumption of 6 ATPs here, 3 ATPs. If there is 1 glucose, so there are consumption of 6 ATPs. So it is an energy consuming process, and the glucose again is now dropped into the blood for reutilization by the muscles and the various other tissues. So this cycle is known as Cori cycle. Very, very important cycle, maintaining the homeostasis of blood, keeps maintain the level of blood sugar. So you can see it here, from glucose to pyruvate, pyruvate to lactate, and then lactate into the blood, into the liver, then lactate is converting to 2-pyruvate, pyruvate to then glucose finally. This glucose again reached to the muscle, erythrocyte, and other tissues like renal cortex, wherever it is being required for energy purposes. Now, a gluconeogenesis from propionate, we'll see it here. Propionyl coenzyme A, which is the major end product of odd chain fatty acid beta oxidation. So there is no fate, there is no utilization of this propionyl coenzyme A. So it is to be converted up cleverly into some very useful product. So this is happening. You can see it here via the enzyme propionyl coenzyme A carboxylase. So wherever carboxylases are there, generally biotin is involved, and there is a consumption of 1 ATP is also there, and carbon dioxide is added to the propionyl coenzyme A, and it finally converts up into the D-methylmalonyl coenzyme A. Then D-methylmalonyl coenzyme A finally isomeracemizes to L-methylmalonyl coenzyme A via racemase enzyme. Finally, vitamin B12 comes as a coenzyme, and the enzyme is methylmalonyl coenzyme A isomerase or mutase. It converts this L-methylmalonyl coenzyme A to succinyl coenzyme, one of the important intermediate of the Krebs cycle. It joins the citric acid cycle or the Krebs cycle, and this is how it can also be involved in the gluconeogenesis. So there is a clear account of how the substrate which are non-glucogenic, how they are involved in the synthesis of glucose during the harsh condition, during the fasting state, or the starvation when we are over with the glycogen resources as a source of energy. So that time the gluconeogenesis is a life-saving and very, very important process. Actively participate in newer synthesis of glucose from the non-glucogenic substances. So what is the clinical significance of gluconeogenesis? The enzyme pyruvate carboxylase. If there is deficiency of this enzyme, and it is a rare inborn error of metabolism as manifested with neurological symptom and mental retardation due to the deficient fuel supply to the nervous system, and accumulation of lactic acid and keto acids lead to the metabolic acidosis. Then hyperammonemia is also seen, and it is an autosomal recessive condition. Then second is ethanol. Ethanol is in excess quantities, it is going to inhibit the gluconeogenesis. You can see how it is involved because ethanol, disrupt the NAD/NADH ratio. So ethanol along with the NAD, it converts up into the acetaldehyde, and NADH is produced, and this NADH is disturbed the whole homeostasis of the carbohydrate metabolism because pyruvate in excess of NADH, pyruvate is diverted to the synthesis of lactate. Oxaloacetate in the presence of NADH is diverted to the synthesis of malate. So excessive ingestion of alcohol result in hypoglycemia, and so lactate also accumulates as an NADH level is high because most of it is diverted to the synthesis of lactates. Glucose alanine cycle, one important cycle, is very much runs parallelly with the Cori cycle. So we already discussed how the glucose in the muscles, the lactate is being accumulated as a consequence of stress, muscle activity. So this lactate or pyruvate being generated, they are also convertible to alanine via the transamination reaction parallelly, and they can produce a huge amount of alanine. And this alanine, via blood transport, it reaches to the liver, and this is actually the nitrogen, movement right from muscle to the liver. So this alanine, it has the nitrogen pool. It is diverted to the synthesis of urea, and this alanine is convertible to a pyruvate, from pyruvate to glucose via gluconeogenesis. Again, glucose is now transported to muscle and other tissues wherever it is being required. So this cycle also parallelly goes on along with the Cori cycle. So converting the useless product to the useful, and how it is involved in the energy production in the form of glucose. So again, I read this line: continuous transport of amino acid from muscle to the liver, which predominantly occurs during the starvation. Pyruvate in skeletal muscle undergoes transamination reaction to produce alanine, and alanine is transported to the liver and used for gluconeogenesis. This is a glucose alanine cycle.